And we can speculate a little bit about, can be very difficult. We can speculate about, and we have no evidence of how life yet started. Okay, given that, there are a number of scientists who propose that in hydrothermal vent systems where we have gradients, so different chemicals coming together, arsenic would have been available for prebiotic chemistry. Whether it was or not, we can't speak to, I wasn't there. But it does chemistry rapidly. It can do chemistry more rapidly. So I think that it will be a fun thing to try and test, but it's not an easy problem.
Randy Shostak, reporter with EOS, newspaper of the American Geophysical Union. How does and might this finding affect NASA's and others' astrobiology programs, including the Mars Science Lab and other ongoing and planned missions and programs?
Do you want me to? Start. All right, I'll start then. With respect to Mars Science Laboratory, or in general the problem of studying habitability on other planets, the way that one could be affected is to think more broadly about what sorts of environments one might characterize as habitable. In specific, with respect to experiments that we could do on Mars Science Laboratory, we do have payload investigations that can measure the chemical elements. We do have a payload investigation that I'm associated with that can actually measure organic molecules. So one way that you can tell what you have is by putting together a number of investigations. And like all science, independent lines of corroboration help you paint a bigger picture. So for example, on Mars Science Laboratory, if you had an environment that appeared to be rich in arsenic or some unanticipated metal, and you also had a finding of organic molecules, you could begin to put a picture together about what the environmental chemistry might portend.
I'll take a shot at that. I think this finding points to a challenge that we've known we've had for a while. Clearly, if we went to another planet or another body in the universe and we saw a human, we would recognize it as life. And there are many other forms we could probably recognize. The challenge of finding something that is significantly different than terrestrial life and life as we know it is really what plagues us when we think about spending the money that we do, developing the instruments that we do, and sending them off on a mission. And so I think it's been our strategy with the support of the scientific community to think as broadly as possible, to put measurements that we may deem reasonable in a context, so make the context of the environment, to explore possibilities that are not so focused on a specific molecule. For example, we would not want to, at this point, necessarily go to look for specifically DNA that had arsenic in its backbone. We want to keep them very broad and keep our strategy in seeking signs of life open to the possibility of things. We would hate to go somewhere and not see it.
Yes, good morning, good afternoon. My name is Luis Fernando Silva Pinto from TV Globo in Brazil. My curiosity is whether the door that you say has been opened will take scientists more in the direction of looking for different environments for life here on Earth or outside. And if you had to pick another element, another medium other than arsenic, which one would you explore first?
So I'll answer the back end of your question first. And I'll tell you, I have a lot of ideas. And we all knew how ideas, let's say, propagated through the nets. And so I'd love to talk to you offline about that. But there's a lot, I have a long career ahead, and I have a lot of ideas I'd like to test. So I think we have lots of questions in terms of other elemental substitutions, other types of metabolism, not just the way you make a cell, but also the way the cells can do their business. So I won't really talk any more about that right now, but I'd say stay tuned for the next 15 to 30 years. Let's say that. That said, the front end of your questions in terms of looking here on Earth or elsewhere, I think yes and yes. I think that we can learn a lot here, not just with my work, but many of the folks, the other scientists supported in the astrobiology program. And again, astrobiology, the study of life in a planetary context. We don't want to be Terran-centric, Earth-centric. Life in any planetary context, or asteroid context, or moon context. I think it's all very important. And so I guess what I'd say is I'm happy to be involved in both of those. And perhaps, Mary, you want to address that? That answers your question.
Right here. Give your name and affiliation.
Sure. Luisa Vilela from TV Report in Brazil. I was wondering, from what I understood, you now open two different doors here, or many more, but one is exploration life as we knew, so expanding the idea of what life is and where it can be sustained. And the other that I was wondering if you could comment a little more is possible practical applications here, like the professor mentioned, the water waste treatments or recovery of phosphorus, or what kind of other possible applications do you see?
I think we're going to direct that to Jim, since he introduced that and is our phosphorus expert. Jim, are you still with us?
Yeah, I'm still here. So yeah, this is all new and spinning out, so a lot of the things just come into your head instantaneously. So, for example, tying together the bioenergy and the phosphorus sustainability issues. So we know we have to begin to develop alternative bioenergy sources for fossil fuels. And so people are growing a lot of plants and trying to figure out a way to grow algae to make bioenergy. But all of the existing plants and algae that we know of need fertilizer themselves. They need phosphorus themselves. So what if someone was clever enough to be able to develop a bioenergy creature, microorganism based on this metabolism, if it holds up, that doesn't need phosphorus. So you don't need to drain the fertilizer supply in order to solve the bioenergy problem. So we're a long way off, but I mean, I would think that this, if this holds up and if we can figure out a way to do it, right, then you can have a whole bioenergy technology that's based on an arsenate type organism. That's kind of science fiction. Probably more realistic are things like treating arsenate waste dumps and this sort of thing to find organisms that are capable of tolerating that or in fact thriving on that, as this one does. So there seems to be applications for toxic waste treatment where arsenate is often an issue. So I think that's another practical aspect that people will start to work on once we start to learn more about the situation. So yeah, it's pretty exciting to think about the possibility of organisms and living things that might be able to live without phosphorus. And for example, just going back to the bioenergy situation, one of the big problems that bioenergy schemes have is that if you make an algae or a cyanobacteria-based bioenergy system with regular organisms, then pretty soon it's going to get infected with other organisms, like it's going to be invaded by things you don't want. Well, one way you could avoid that is that all those other invaders are probably going to need phosphorus, and so if you had a phosphorus-free environment with a bioenergy organism based on arsenate, instead, that's going to be pretty resistant to being contaminated by invaders that you don't want. So I think, there's pretty exciting stuff here if this holds up.
I could also comment, I think, on the end of that, Jim, and that, so if this microbe has been something we knew, so when I said it looked ordinary, also what it does is very ordinary. So you and I breathe oxygen and we burn sugar. Same thing this microbe does. So metabolically, scientifically we'd say metabolically, it's not very interesting. It's something very normal in terms of its metabolism. And the way it looks, again, we would look in the environment. We may not know that one thing, it wiggles, it moves really fast, they swim quite rapidly. We may not notice that, that microbe is making its biomolecules out of some other element. So if for every one of your cells in your body right now, there are 10 microbial cells. So you are mostly microbe. What might we not understand about those microbes? So that's in you. Now let's go global. So on planet Earth, we know that it's very well supported now that microbes are some of the major drivers of the biogeochemical cycles on Earth, like carbon cycle. So if microbes are these major players in our own bodies and in the Earth, so on our whole planet, if we're missing a population maybe based on arsenic, but maybe these other possibilities that we don't even know yet. I think it has vast implications in understanding the way our own bodies might work and help inform us about that, but also in the way our planet works. So that I think Jim suggested in practical applications that it's absolutely, but some of the practical applications will develop over time to understand, here's the fundamental discovery, And, you know, it's going to take an army of scientists, clearly not just myself and my team, but other people to bear on this problem with their tools and their ideas.
I think it's clear from both of their answers that we're opening up all sorts of new areas of research. Jim, you're practically breathless with your ideas. Well, I'm sure you'll be looking for money soon. So, you know, the ramifications of this finding are pretty significant.
Before we go to the West Coast, we have some time for the gentleman here. You can give your name and affiliation, sir.
Sure, it's Ivan Semenik with Nature. I just have two very short technical questions, non-technical questions, just detailed questions. I'm wondering about the fraction of the swap out. The video kind of implied all the phosphorus molecules disappearing, or atoms disappearing, being replaced with arsenic. Do you have a sense of whether it's that complete or whether it's some fraction, and if there's some level, some threshold there? And for Steve Benner, if you could just clarify what it is about arsenate that makes it a weaker link.
Yeah, I mean, I'm just trying to understand what the level of explanation should be, how the explanation should be crafted to meet the level at which you're interested in an answer. Arsenic has, of course, a position below phosphorus on the periodic table that has been mentioned. Because of that, it has orbitals, which we chemists like to talk about, d orbitals, f orbitals, that are lower in energy, and that permits arsenate to fall apart by what is called in the business an associative mechanism. That is, the attacking water doesn't have to wait until something leaves in order to attack. It attacks without waiting for something to leave, and that's quite different than what molecules farther up in the periodic table do. And so this is why, again, my curmudgeon wet blanket rolls, I sit here and say, it's wonderful to say that we're going to go to other planets and get a phosphorus-free energy. Right? What we believe, based on prior experience, is that what will certainly survive from Felice's work is the microbe. And that microbe, if it grows in low phosphorus environments, that's wonderful. I mean, that can be used practically. But we don't believe right now that the body of evidence saying that that molecule can't exist at the present time overwhelms the body of evidence that says that molecule does exist. And that's not a theoretical argument, that's an argument based on empirical data. I'm sorry, yes.
Back into that, and maybe I should have answered first. And it's true what Steve's saying. I'd like to let him
get the wet blanket
off. So, disposing with the wet blanket, what I will say is what we presented, so that was an artist rendition, absolutely, to illustrate the point. If you're not so familiar with